Assalamualaikum warahmatullahi wabarakatuh. We are from Group 3. Will present about viruses and nematodes as biological control agents. Before starting the presentation, I will introduce our group members. The first, Aida, Femnin, Kamalia with student number 073. Second, Anggito Dewanto with student number 074. The third, Arifa Nur Hanani with student number 081. And the fourth, Choirin Wahyuning Tyas with student number 082. Bionomy of viruses as insect pathogens. Viruses can cause disease in insects and mites. Viruses are divided into six groups. The first, Baculovirus. The second, cytoplasmic polyhedrosis virus. The third, Entomopoxyvirus. The fourth, Iridovirus. The fifth, Dewanto, and the sixth, small RNA virus. Baculoviruses initiate an infection cycle in much the same way as other large DNA viruses, and employ transcription and sensor and activators to exploit the transcriptional host apparatus. Baculoviruses are the only viruses that replicate nuclear DNA from eukaryotes that use a combination of cellular and viral polymerases.
Next is cytoplasmic polyhedrosis viruses, or CPVS. CPVS collectively form a genus within the Rheoviridae, whose host range is apparently confined to the arthropoda. Unlike other members of the Rheoviridae, which have a two-layer capsid structure, CPV particles have only a single-shelled capsid and are in many ways morphologically and biochemically comparable to the virus course of other reoviruses. Since CPV particles don't possess an outer capsid layer, they arguably don't contain proteins that are comparable to the outer capsid proteins of other members of the Rheoviridae. Next. Entomopoxyvirus. Entomopathogenic viruses are microorganisms that attack insect pests and make them sick. The known types of entomopathogenic viruses are isolates from SIMPV and HANPV. Entomopathogenic viruses are obtained from dead insects that are infected with the virus. Insects that die from infection with viruses that can be found on the surface of leaves, stems, or other organs of plants. Its characteristics are dead, rotting, and black larvae. Next. Iridovirus. Iridovirus is a DNA virus classified into the family Iridoviridae with a hexagonal or icosahedral shape when viewed through an electron microscope with a diameter of about 120 till 240 nm and develops in the cytoplasm of infected cells. Iridovirus can cause death within 24 till 48 hours after the appearance of symptoms of infection. Iridovirus is a disease DNA virus that causes systemic infections in freshwater aquaculture and seawater fish farming. Next. Densovirus. Densovirus is a mutualistic symbiont of a global corpus Helicoverpa armigera and protects against Baculovirus and biopesticide. Densovirus is mainly distributed in the fat body of insects and can be transmitted horizontally or vertically.
Small RNA virus. Small RNA regulate a large set of gene expression in all plants and constitute a natural immunity against virus. Small RNA-based genetic engineering technology has been explored for crop protection against virus for nearly 30 years. Viral resistance has been developed in diverse crops with SRGE technology, and a few viral resistance crops have been approved for commercial release. Based on their morphology, pathogenic viruses are divided into two groups. First, viruses that have inclusion bodies protein, such as packaged Baculovirus, nuclear polyhydrasis, granulosis virus, cytoplasmic virus, CPV virus, and Entomopoxyvirus. Second, virus lacking protein inclusion bodies such as Iridovirus, Entomopoxyvirus, and small RNA virus, and unwrapped Baculovirus. Transovarial transmission of the virus, insect pathogenic virus enter the mouth to the digestive tract. Viral infection occurs through the intestinal epithelium or into a susceptible host. Certain viruses only attack specific network, while other viruses don't attack the entire network or polytrope. Insects infected with the virus will show physiological, morphological, and behavioral symptoms. Symptoms that appear will vary depending on the type and host. The next topic is entomopathogenic nematode BIONOMY.
2. Entomopathogenic nematodes BIONOMY. Entomopathogenic nematodes are insect parasites that live in the soil. The term entomopathogen, entomo comes from Greek words meaning insect and pathogen meaning causing disease. Penetration of NAP is done directly through the insect cuticle and natural folds such as spiracles, mounds, and anus. Entomopathogenic nematodes have several advantages, namely they are virulent to their host, kill host insect quickly, have a wide host range, and are not harmful to non-target insect, and they are easily cultured in vivo and in vitro. Entomopathogenic nematodes, or NAP, are biological control agent from stem family. Nematodes kill insects through the mutualistic symbiotic process with bacteria carried in their digestive tract. The relationship between bacteria and nematodes is mutualistic because both benefit. Although nematodes can kill a host insect in the absence of bacteria, infection is slow and cannot reproduce without eating the bacteria that supply nutrients. NAP life and reproductive cycle. Entomopathogenic nematodes have a simple life cycle and have the main stage of development from age juvenile and adult. Juvenile is divided into juvenile instar 1 or G1, juvenile instar 2, G2, juvenile instar 3, G3, and juvenile instar 4, G4. The life cycle of nematodes from infecting until the emergence of a new generation of juvenile range from 7 until 10 days. Juvenile leave the carcass of the host 2 until 3 weeks after developing in the host body and looks for a new host. The change of instar is marked by a period of molting. So the reproduction of NAP continues until the surge of nutrients in the host body is exhausted. Infective juvenile leave the host and search a new host. Infected juvenile can survive without a food for several months until they find a new house. So that's it for our presentation. Thank you for your attention.